Modern drug discovery is a massive logistical challenge, requiring scientists to find where a new medicine physically bonds with a shifting biological target. To determine if a drug candidate will actually work, researchers must model that physical connection at the atomic level. But human biology is fluid and chaotic, while traditional supercomputers operate on a rigid architecture. They process information strictly in binary ones and zeros. Atomic interactions inside a protein do not follow those binary rules. They are governed entirely by the complex probabilities of quantum mechanics. Because our most powerful computers don't natively speak the language of biology, they inevitably hit a computational wall when trying to map out detailed medical designs. The logical solution is to use a machine built to handle those rules, a quantum computer. These machines offer a potential advantage simply by operating using the same quantum mechanics as the biological molecules they are trying to simulate. But there is a catch. Current quantum hardware is fragile and easily disrupted by background computational noise. Because of this interference, early quantum processors were easily overwhelmed, typically maxing out at simulating tiny molecules containing only a handful of atoms. We built a machine with the right vocabulary, but it lacked the stability to read the tens of thousands of atoms required to accurately model a human protein. That bottleneck was recently bypassed through a collaboration between researchers at IBM, Riken, and the Cleveland Clinic. This chart tracks their milestone. In 6 months, the team scaled up quantum simulations by 40 times, jumping to the 303-atom trip cage protein. Pushing further, they modeled the 11,000-atom T4 lysozyme and finally the trypsin enzyme, calculating the energy of a massive 12,600-atom system. At this scale, a molecule is a dense, highly complex biological machine with thousands of interacting variables. Hitting this benchmark proves that simulating large, clinically relevant proteins is a practical reality rather than a theoretical goal for the distant future. So, how exactly did scientists calculate something this massive without inventing a completely flawless, perfectly stable quantum processor? They used a workflow called quantum-centric supercomputing, a hybrid tag-team strategy that divides the labor between classical and quantum systems. First, a traditional supercomputer chops the massive target protein into manageable fragments. Next, the quantum processor calculates their behaviors, keeping the data load small. Finally, the supercomputer reassembles those outputs to form a highly accurate, complete picture of the molecule. By dividing the work, this hybrid strategy bypasses current hardware limits, letting each type of machine focus on its specific strengths. This framework is already moving past computer science experiments to target specific diseases. Recently, a software team called Algorithmic used this hybrid method to model how experimental cancer drugs can be activated by light to destroy tumor cells. In genomics, researchers at the Wellcome Sanger Institute successfully translated the entire DNA sequence of the hepatitis D virus into a quantum-readable format. Their long-term goal is to use this methodology to map and analyze the full human genome. These early applications are pulling quantum computing out of the laboratory and turning it into an actionable toolkit for medical research. Even with these strides, true quantum advantage, where a machine runs entirely on its own without any classical assistance, has not yet been achieved. The underlying hardware is still constrained by physical instability and system noise. However, by using this hybrid approach to sidestep those issues, experts expect the technology to mature into a practical scientific tool by the early 2030s. By learning how to translate the language of biology into computable data, scientists are finally building the machines required to understand and cure the human body at the atomic level.